So I'd like to introduce the first speaker, which is Dr. Terry Watnick from the University of Maryland. Yes, you know, Dr. Watnick is a clinician scientist who also directs the PKD Center at the University of Maryland. And she's really a true expert and is going to tell us about the PKD pathobiology that would be relevant for clinicians. And thank you very much, Terry, for taking your time. And I know you're very busy schedule.
Just recently.
Yeah. Okay.
Okay, thank you. Okay, I just want to thank the organizers for inviting me, and thank you to Dr. Pei. It's always a treat to be here, I think, especially at this time. So I'm going to be speaking about PKD pathobiology. What do clinicians need to know? I don't know if you need to know all of this. I think it's quite a big task, and I just want to say that I'm probably just scratching the surface here, and I hope I can do it justice. So here are my disclosures. And so here is what I hope to do today, which is to update the key concepts on PKD pathobiology, highlight some of the cyst growth modulation pathways in PKD, and then talk about metabolic reprogramming because a lot of therapies now are focusing on that. As you guys all know, ADPKD is the most common genetic cause of end-stage kidney disease. It is common, affecting 1 in 500 to 1 in 1,000, regardless of what country you come from, what ethnic background. And as you can see there, it's characterized by progressive development and enlargement of kidney cysts, and that's what an end-stage PKD kidney looks like after this has gone on for a number of decades. About 15% of people will go on to develop end-stage kidney disease. And although we as nephrologists treat the kidney complications, this is, there are many extrarenal complications, including polycystic liver disease, which we're going to hear about this afternoon, and brain aneurysms. So this is important to know as a clinician, which has to do with the genetics of PKD. It may be covered as well in other talks, but about 85% of cases of autosomal dominant PKD are due to mutations in the PKD1 gene, which is on chromosome 16. It's a very large gene, a very large protein, as you'll see. The protein we call polycystin 1. About 15% of cases are due to mutations in PKD2, which is a smaller gene, and we call that polycystin 2. And then I would say that over maybe the past 5 to 10 years, we have come to appreciate that there are mutations in a number of other genes, which as a clinician you might confuse for PKD. A lot of those were identified at Mayo Clinic by Dr. Torres and his colleagues. And so it's just important as a clinician to be aware of that. If you look at enough scans, you can see that they're not exactly the same, and maybe Dr. Klein will cover that, but important to be aware of. As a clinician, you know, you're going to probably take a family history, and one thing that you will notice is that those individuals with PKD1 mutations tend to have more severe PKD compared with those with PKD2, and a lot of people have looked at this, but I don't know how to work the pointer exactly, but you can see in the chart there, the red is the cumulative probability of renal survival in those individuals with PKD2. And you can see that the curve is moved to the right, and that's in comparison to those with PKD1 mutations that are truncating, meaning that there's probably no protein made in the blue, and you can see that those people develop end-stage kidney disease almost 2 decades earlier than those with PKD2. And then within the PKD1 class, you can break that down further. There are those individuals who have missense changes, or I guess another way of putting it would be you swap one amino acid for another. And some of those may have partial function, they may not. It's hard to say, but as a group, those individuals are kind of halfway between those individuals with PKD1 truncating and those with PKD2, and that's indicated by the green curve there. Okay, so what about polycystin 1? We're going to get into a little bit of biology here. As you can see, it's a very large membrane protein that resembles a receptor. So it has 11 transmembrane domains. I don't, where's the pointer exactly? Oh, okay, over there. right here, the 11 transmembrane domains. There's a large extracellular portion that sticks outside the cell and a small intracellular portion. Okay, as it turns out, and I'm not going to get into this too much, a little bit at the end, it turns out that proteolytic processing of this big membrane protein actually turns out to be important for its function. And probably the most well characterized of those is this one right here, this GPS cleavage site. If you mutate that, you can wind up with kidney cysts. So that process is very important. So this is a very big protein. It's got numerous motifs, and there have been many, many proposed functions for this protein, and I've listed those there. As a flow sensor, I'll get into that, an atypical G protein coupled receptor, et cetera, et cetera. So I think it's fair to say we still don't completely know what the function of this protein is. Polycystin 2, shown there, is also a membrane protein, and it's thought to be a member of the transient receptor potential family of cation channels. Basically, it's a channel. It has 6 transmembrane domains. It is a calcium permeable channel, but actually, as I'll touch on later, it's not so clear because this channel is also permeable to sodium and potassium, and thought possibly to participate in flow sensing, although there's kind of conflicting data in that regard as well. So putting it all together, these two proteins actually interact with one another. In other words, they bind together in the cell, and they form what we call a receptor channel complex. So it is thought that when these two proteins work together, they mediate calcium-dependent signaling pathways. Okay, and I would say that the upstream and downstream signaling pathways that activate this complex are still not completely understood. The fact that these two proteins bind is important in a clinical sense, which is because they're so central to the signaling pathway, whether you mutate one or the other, you essentially get the same disease. So, okay. So I think the question that York wants me to answer, which I'm not sure I can completely answer, but we'll try at least to give you an idea of where we stand, is how do you go from this normal kidney to that? And that kidney weighed 40 pounds when it was removed from a patient of mine. So how do you get from that? Obviously, it's a complicated process and it takes many decades. This isn't something that happens overnight. So one thing that we have come to understand, and I guess it does have important clinical, it affects clinically, is when you look at an early stage kidney, so this is a microdissected kidney from an early stage PKD kidney, cysts are forming in certain places, right? It's not forming throughout the tubule. So you can see a cyst here, 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 and they're of different sizes, suggesting that they may have kind of started at different time points. And in between those cysts, the tubules appear pretty normal. And so we do have some understanding as to why that happens. When I was a fellow, we actually undertook, and this is in collaboration with York actually, genetic analysis of individual cysts. And what I'm going to show you here is kind of a summary of what that all showed. So as you know, when you're born with PKD, you inherit the same mutation in all the cells in your body, okay? So, and that you have 50% because it's an autosomal dominant disorder. So you have one mutant copy of your PKD gene and then a normal copy. And that's in every cell. But what we know happens as a result of that, all of that genetic analysis we did, is that you have a somatic mutation of the wild type allele. So that's the gene that you inherited from your unaffected parent. And this is kind of happens randomly. Once you do get that, you wind up with a cell that has no normal PKD gene in it, and you have clonal expansion of that PKD deficient cell, which then over time results in tubular dilatation and fluid secretion, leading to a cyst. What we know, and I'll show you some more data in this regard, is that because this is happening randomly and stochastically, every cyst contains a unique somatic mutation. Okay, so even though you're born with the exact same mutation that you inherited most of the time from your parent, what happens in that unaffected gene is each cell will be different. And so what this means, what this fundamentally means is that PKD is recessive at the cellular level, or that you're losing functioning of the complex, because that's not always the case in all autosomal dominant disorders. But in this case, it's because you're losing protein function. Okay, and so I want to say that other people, this is kind of a little bit controversial, although I think the data supports that this is a major mechanism, is that there could be other processes that result in decreased polycystin signaling. I think mutation of the somatic allele is the most common, but it doesn't have to be the only one. So anything that lowers polycystin function could result in a cyst. So the question is, how often do you see these somatic mutations occurring? I did a study with York many, many years ago, 2 decades ago now, and using maybe not state-of-the-art genetic methodology at that point, we were able to find somatic mutations in 70% of cysts. But more recently now, with whole genome and whole exome sequencing, you can really do quite a bit better. And this is a study from the group at the Rogerson. They actually screened 90 cysts from 24 different donors. 21 of them had PKD1, the remaining 3 had PKD2, and they used whole genome sequencing and were able to identify unique somatic mutations in 93% of the cysts. So that's quite high. So I think we can conclude from this, and this is just showing some of their primary data on the right, showing that all of these mutations are indeed unique. I think we can say that in human ADPKD, somatic inactivation of a PKD gene is probably the primary cause of cyst formation and expansion. May not be the only one, but it's probably a major cause. Okay, so that's probably the first step in going along that pathway. You have to have loss or decrease in polycystin signaling, but then what happens after you get your somatic mutation? There's kind of a lot of steps in between there that I think we still need to understand. And this is from a paper by Vicente Torres, looking at all of the effector pathways that have been implicated in PKD. And you can see that this is quite complicated, and there's a lot of stuff there, and I don't think that I can probably cover all of it in this short talk, but I will be focusing on a couple of different pathways that I think have kind of maybe risen to the top as being quite important, and those are in red there. So I'm just going to say a little bit about cyclic AMP, and again, this is from a review by Vicente, who is the expert on this, but I'll summarize it. I think a lot that we have come to understand that cyclic AMP may be a major modulator of polycystic kidney disease. And so the thinking is summarized in this cartoon on the right, and I'm going to try to summarize it. So I told you that polycystin 2 is a calcium channel, or at least calcium permeable. So the thought is if you mutate all of that, you have a disruption in intracellular calcium homeostasis. And the levels of calcium in the cell are completely critical to so many functions of so many different proteins. So if you mess with that, you can just imagine that you're going to disrupt a lot of stuff. But one of the things that you could disrupt is cyclic AMP. And the reason for that is that adenylate cyclase, which is the major hormone that makes cyclic AMP, is calcium sensitive. So by decreasing calcium, you might increase adenylate cyclase and increase cyclic AMP production. Also, some of the phosphodiesterases that break down cyclic AMP are also calcium sensitive, so you might inhibit those by lowering the calcium in the cell. And why is cyclic AMP level so important? Well, for one thing, it stimulates protein kinase A, which is shown right here. Okay, so and it stimulates protein kinase A to phosphorylate proteins that result in proliferation and fluid secretion. Okay, so that's the idea. And cyclic AMP, one of those very important proteins could be CFTR, which is postulated to be the apical cyclic, it's a cyclic AMP stimulated chloride secretion, which then drives fluid secretion into cells. And as it turns out, and this is really important, is that vasopressin, or antidiuretic diuretic hormone, is the major hormone in the kidney that has cyclic AMP as its second messenger. So if you increase cyclic AMP levels, you're going to stimulate all of that, which is then also could promote all of these bad things, including cell growth and fluid secretion. So that's the basic idea. And it has turned out that this is very important and very important clinically. And I don't know if anyone else is going to cover this, but there's extensive preclinical data, a lot of it from Vicente's lab, supporting a role for vasopressin and cyclic AMP and PKD pathogenesis. And I've just pulled out from a bunch of papers some of that data. Right here, you can see this is the PCK rat, which is a model that people use for PKD. If you cross that rat that has PKD to a vasopressin mutant rat, you can see, you really abrogate cyst formation. And here in another study, they mutated adenylate cyclase 6, which is adenylate cyclase in cilia, which I'll get into. And if you combine that with PKD, here's PKD mutant kidney, here's a wild type, and you can see if you knock out this adenylate cyclase, you make it better. And then again, this is really kind of seminal data from Dr. Torres's lab, looking at a whole bunch of PKD mouse models. And if you treat these with a blocker of the V2 receptor, you can improve cyst formation. So all of this very kind of important basic science data led to this paper, which I won't talk about much, only to say that there have been many, many studies looking at this pathway and different kinds of approaches to blocking it. So this has turned out to be critical, and I'm going to leave it to everyone else to talk about it more. And move on to the next topic, which is the role of cilia in PKD, which it's turned out to be quite important as well. And for those of you who are clinicians, you might wonder, well, what are primary cilia? It turns out that cilia arise from the surface of most cells in your body. And there I've just taken from the literature all different kinds of cells showing that primary cilia are there. At one point in our scientific evolution, these were kind of thought not to be very important organelles, but it actually turns out that they're critical, and they organize various signaling pathways, which I'm not going to get into, but it turns out developmentally is very important. If you mutate that, you wind up with kind of a bunch of different abnormalities. And it's thought now that these cilia organize signaling pathways and act basically as chemo and or mechanosensors, so very critical. And it also turns out that these cilia are central to the pathobiology of PKD. So this polycystin complex that I told you about, polycystin 1 and 2, localized to the primary cilium, along with all kinds of other genes that when you mutate cause cystic kidney disease. And that's just a cartoon from an old review that we did showing that all of these genes are somehow associated with cilia. KIF3A is a protein. It's a subunit of kinesin 2 motor complex that's required for cilia formation. And this is just taking one example to show you. And if you mutate that gene, this is just showing the KIF3A mutant kidney, you get cilia. So you get cysts. So if you don't have cilia, you wind up with cysts. Okay, so this is just, I guess, updating some data. So what you would think, or what I would think, is that if I mixed one of these mutants with PKD, I would get much worse cystic disease. And it actually turns out that that's not the case. Okay, so here is your PKD kidney. These are mouse models, and I'm not going through all the details, but this is a mouse where you have PKD because PKD1 has been deleted. And here is no cilia because of KIF3A. And you can see that it's cystic. It's not exactly the same, though, right? Because this kidney isn't as big as that one. But if I combine these, I'd think it'd be worse. But actually, it turns out that it's better. So in this kidney where you've deleted PKD1 and you also have no cilia, you can see that this is not as bad as that, right? I mean, that's pretty clear. And so the model is, it's a little bit complicated, but the model is that when you have a cilium and you have the polycystin complex in the cilium, everything is cool, no cysts. However, If you do this, you get a cyst, and it's because there is something in this cilium that polycystin is repressing. And when you lose that repression, you get a cyst. If you knock out this cilia, well, then you don't have that pathway there to repress, and that somehow makes it better. This is work from Steve Somlo's lab. It's called cyst-dependent cyst activating factor. And I didn't have time to add this in because it's just really brand new, but he's gone a long way to trying to identify what that CDCA factor is. It turns out at least one of them is a transcription factor called GLIS2. And I won't get into it, maybe next time. So now I guess we can come back and say that might be one thing that the polycystin complex is doing in cilia, but another hypothesis has been that it regulates luminal flow, and that thus is regulated by luminal flow and controls tubular morphology. So the idea behind this is that polycystin 1, which is that sensor, is sensing flow in the kidney, right? Because your urine is going by those cells and there's flow there. And so the idea is that this fluid flow bends the cilium, and this results in calcium influx. And polycystin 1, that sensing of the flow, is what stimulates this calcium influx. And when you have this in the cilium, this then would suppress growth, induce proper differentiation and maintenance of your kidney tubes. So that's the idea. I think it kind of is going to wind up being a lot more complicated than that. And I just love this. So this is a little bit of a complicated biology, but I do think it's cool. So people actually were able to look at that hypothesis by developing a way to measure calcium in the cilium, which is, I think this is cool. Let's see if it's going to work. Oh, the movie didn't work. Well, there is a movie here that shows them with this little patch pipette attaching onto the cilium. Oh, is it working? Oh, let's see. It's not working. Okay, well, it didn't work. It shows that it's pretty cool. I think this is just, it's just a great demonstration of how kind of very technological advances can lead you to be able to answer some pretty complicated questions. But what they were able to show, if you grow a cell in a dish and you patch onto this cilium, and you can measure calcium influx that way. If you use a way to knock out PKD2, you actually do decrease the amount of calcium in the cilium. Okay, so that is true, and you can see it here. You can use you can block all these other different proteins and there's no effect. But when you block PKD2, you can see that's the case. And then this is looking at mouse cells. And it's the same thing. If you knock out PKD2 in mice, you can see that the calcium influx is less, and that's not the case with PKD1. So that part of the model maybe is true. But the question is, does flow actually cause an increase in calcium influx? And I think that part is not so clear. So again, this is just a real technology feat to encode calcium indicators into the cilium so you can directly look at this. And I'm not going to get too much into the weeds, but I do find it kind of cool that they took this tube from a mouse kidney, you know, the actual tube, and you can see it here, and there's the little pipette. And they forced flow by this and then looked at calcium influx. And you can see here that they're actually deflecting the cilium, but when you look at calcium, it isn't changing at all. So even though I think it's fair to say that maybe the polycystins in the cilium do control calcium influx, whether this is flow related or not, I think is not like quite as clear. So just quickly again, and I don't want to get too much into the weeds, but this is again a demonstration of how all of the most state-of-the-art technologies have been applied to our field, which is, I think, really, really important. And this is a new, I guess, technique, cryogenic electron microscopy, where you use that to look at protein structure. And so what they've done here is look at the polycystin 2 structure. And it turns out that polycystin 2 all by itself can form a channel. And there are 4 different polycystin molecules, and they form a channel all on their own. And you can see it here. These red things, these little red dots, indicate negatively charged amino acids, which is going to make positively charged go through, or calcium. It also turns out that you can get polycystin 1, polycystin 2 combined channel. I didn't get into it, but polycystin 1 has some channel features to it. And here, in this case, you actually have a negatively charged residue, so you wouldn't expect calcium to be able to get through that. So the real question is whether this is negative inhibitory and that's positive. It's just so hard to say. It's complicated, and I think that a lot more work needs to be done. And all I want to conclude is that the relationship between the polycystins and intracellular calcium homeostasis is quite complicated. And I think a better understanding, however, is fundamental to unraveling the pathophysiology. So the great thing is, from the PKD standpoint, and I want to emphasize this because I'm part of this consortium that focuses on this, is that we really do need to have the best technology from across the science field focused on this, because we don't know what advances are going to be made. So we need to continue this. Going to switch to metabolic reprogramming in PKD, which is going to be the last major thing I cover, which is, so this comes from an observation. It's a very simple observation that when you grow PKD mutant cells in culture, there's more acid produced. So, you know, when you grow these cells in culture, you're growing them in a media that looks like it's got this color, but it contains a pH indicator. So when it gets acidic, it kind of gets this orangey-like color. And so people noticed that this was kind of different, that you grew normal cells in culture, they looked one way. You grew PKD cells in culture, they looked another way. So if you look at that a little more carefully and see what is in the media of these cells, you can see, and that's shown here, that there's increased lactate, right? That's probably what's making the media more acidic. And on the opposite side, it turns out that the cells are using a lot more glucose. So if you look at glucose, you can see it's lower. That's this black line in the mutant cells, whereas lactate is higher. And then if you look at glycolysis and the enzymes that are required for glycolysis, you can see that they're very upregulated in these mutant cells. So this was a simple observation, but it's led to a whole array of downstream studies. And I'm just going to touch probably on the surface of this. But basically what this gets to is that PKD1 mutant cells are depending on aerobic glycolysis for energy. Okay, so this is getting back to some basic biology. But if you remember, you know, just any old cell is going to metabolize glucose to pyruvate, right? And that's shown here. You know, and when oxygen is plentiful, you know, you have oxygen around, pyruvate is going to be completely oxidized to carbon dioxide via the TCA cycle and oxidative phosphorylation in mitochondria. I don't know if you remember that. And that's shown here. However, when oxygen is limiting, you know, like when you exercise and you know how your muscles hurt and all that, that's because you don't have oxygen around, and instead glucose is going to pyruvate and lactate, and that would be anaerobic glycolysis. So cancer cells, it turns out, have adapted this and instead convert most of the glucose to lactate, regardless of whether you have oxygen available or not. Okay, and that's shown here. This is called the Warburg effect. And you might wonder why a cancer cell would even do that, because you need all of this ATP, and the other way you get 36 molecules of ATP, whereas this way you only get 2. So why would you want to do that? And the reason is because this is actually turns out to be anabolic, and there's all kinds of intermediate metabolic products that are used for the cell to grow, and it can happen very quickly. So just to summarize that idea, metabolic reprogramming provides carbon for the production of building blocks, nucleotides, lipids, proteins, et cetera, which new cells need to grow. So in some ways, this is getting to this idea that PKD has some similarities to cancer in terms of metabolic profiling. So And you can, they've gone on to show that Alessandra Balletta's lab that if you inhibit this process, you actually can make PKD better. And the way they did this was using 2-deoxyglucose or 2-DG. And this is a glucose analog, okay? But it can't be metabolized. So the cell will try to use it, but you can't get to that downstream pathway that I showed you. And it turns out that if you treat mice with 2-DG, you can improve PKD. And I won't go through all of the data, but just to show quickly here, this is a PKD mouse. These are cystic. This is the control. And if you treat them with 2-DG, they are somewhat better. Looking here at the total kidney volume, you can see this is statistically significant. There's some decrease in the total kidney volume. And then again, looking at MRIs of mice treated with 2-DG, at 2 months, you can see that there's fewer cysts. So this pathway, or if you can sort of work on this metabolism thing, you can make it better. Just going to quickly touch on mTOR signaling, which is, again, part of this whole metabolic pathway. The idea that mTOR might be involved in PKD is kind of a little bit of night move thinking, but people realize that there are a subset of patients with tuberous sclerosis that actually have cystic kidney disease, and you can mistake it for PKD. So this led to the idea that there was some relationship between that. And this is, of course, again, a really complicated pathway, but I just want to briefly summarize it to say that mTOR, which is the central protein in this complex, is an energy sensor. And the genes that are involved in tuberous sclerosis actually negatively regulate it, meaning that when they're intact, they suppress it. And there are tons of inputs on this pathway. It's like super complicated. The bottom line is, though, that there are some that inhibit mTOR and some that activate it. And the one that I want to focus on is this AMPK, okay, which the higher it is, it inhibits this growth system, okay? And because I'm running short on time, I just want to very quickly say that it turns out that when you look in cells, AMPK is down-regulated in PKD. So that when it's intact, it's a negative regulator of growth. If you decrease it, it's going to mean more growth. So, okay. So metformin is a drug that increases AMPK. And in mouse models, it turned out that this had some positive effect. And that's shown here in 2 different mouse models. There's some improvement. Cystic index decreased by 52%, from 71 to 52 and 43 to 31. So in mouse models, this had some positive effect. How this might work, in addition to inhibiting mTOR signaling, it's thought to inhibit the activity of CFTR again and activate phosphodiesterase, decreasing cyclic AMP. And this is just shown again in a pig model of PKD, that it's quite effective. So here they're looking at a control pig kidney, treating with 2-DG, which we talked about in mice works, metformin works, and when you combine them, actually, it doesn't look to be like there's an additive effect. My last metabolic reprogramming topic has to do with fatty acid oxidation, which it turns out is reduced in PKD. And I just want to say again, this is kind of like the general pathway for ATP generation in the cell. And as you remember, this is the TCA cycle, but you can get into this TCA cycle by using fatty acids, right? So if you break down fatty acids, you're going to get to acetyl-CoA, and you're going to go through this whole pathway. And it turns out that fatty acid oxidation is reduced in PKD. Okay, and this is work from Greg's lab. These are cells, and the dotted lines are all the PKD mutant cells, and in every case, they're lower in terms of fatty acid utilization compared to the wild-type cells. And if you look, again, this is work by a different group. If you look at PPAR-alpha, which is a transcription factor that controls a lot of the proteins that are involved in fatty acid oxidation, that is decreased in PKD mutant mice. So you can increase that by fenofibrate, which is a drug that I don't know if you guys use so much. We use it occasionally for people with hypertriglyceridemia. And you can see that if you treat mice with fenofibrate, PKD mice, that you can increase the level of this PPAR alpha. And again, you can treat mice with this, and it seems like there's some improvement. So, you know, all of these kinds of processes are going on in mitochondria. So the question is, what's the relationship between PKD and mitochondria? And again, this is work from Greg's lab, just looking at the mitochondria and PKD mutant cells. This is electron microscopy. And you can just see, if you look at this versus this, that these are kind of normal mitochondria and these are kind of not normal. So there is some relationship here. And again, getting back to, well, what is the relationship? I'm going to not go through this in the interest of time, but I want to say that a lot of people are now looking at the very C terminus of polycystin 1. Remember, I told you that there's all these little pieces cleaved off of it? Well, a lot of people now think that the very end, this little piece here is cleaved off and goes to mitochondria. So what the function of that precisely is, I think we really don't know, but a lot of people are working on that. And I just want to end by saying, I told you that this kind of observation, a very simple observation in cells, has led to kind of an explosion of different ways to try to test these kinds of things. And this is just a summary from a review of all of the interventions that have been tried that are aimed at this issue of metabolic reprogramming. So it's definitely something to watch. I don't think we're done with this yet. So I just want to say in summary that PKD, you get PKD when polycystin function falls below a critical threshold. Most of the time in humans, that's going to be by a somatic mutation. PKD proteins are part of a very complicated nexus of signaling pathways. Many of these pathways are interrelated and involve positive and negative feedback loops. Cis modulation factors include cyclic AMP, which we went through, calcium signaling, and cilia function. I think a lot of work has gone into showing that altered metabolism is a major driver of PKD. And I've listed some of that there, some of the factors there. We have enhanced glycolytic flux, which we went through, increased mTOR signaling, decreased AMPK, decreased fatty acid oxidation and utilization, altered glutamine metabolism, which I didn't get into. And then I think there's still a hope that targeting these metabolic pathways could modulate disease progression. And there are many, many ways to do that. And I think we're in the midst of kind of testing some of that. So I'm going to end there.
Thank you very much, Terry, for a very comprehensive review given the time restraint. Any questions from the audience? Yes, hi, Stephen.
Just one going back to the original.
Can we get the mic for him?
Just going back to your original report about the somatic mutation, just to be clear, if you've inherited PKD1 from your mother and have an unaffected chromosome from your father, you're confirming that there has to be a somatic mutation in the paternal gene as well to activate the maternal abnormality?
50% of the protein, and 50% of the protein, like if what you inherit is good. You can probably live and not do probably pretty well with that. And that's been shown in mice. You know, if you have a heterozygous mouse with one mutation, that mouse is fine. You might get an occasional cyst, but pretty much they live normally. So, but it's when you mutate that other allele. So,
and each of those mutations in each cyst is different.
It looks like they're different.
Excellent. Do you have any data on, you compared PKD1 to PKD2, any data on the frequency and results of patients who may have both mutations in both? I have one patient who has both PKD1 and PKD2 mutation.
Yeah, so I mean, I think there's been data showing that those people will do worse, like compared, I guess presumably they inherited one from one parent and one from the other.
Perhaps, yeah.
Okay, so
I
there's
haven't
been
tested
some,
the parents.
okay, well, let's assume.
Yeah.
So, you know, that they would probably do worse. That would be the idea, because you could have a somatic mutation in either gene, so that would increase the rate of that happening. I would
Terrific. Thank you very
guess.
much.
I mean,
Thank you very much, Terry.